Welcome and thank you for attending our workshop, Advances in Synthetic Oligo Synthesis to Revamp Your Workflow. My name is Ybert and I'm one of the co-founder and the CEO of DNAscript. Last year, we reported the feasibility of enzymatically synthesizing 280 nucleotide oligos to demonstrate the remarkable capabilities of our technology. Earlier this year at LGBT, we shared more progress on the development of our technology. And today I'm tremendously excited to tell you about our latest achievement, which is the commercial launch of our first product, the Syntac system. I also want to thank our guest speaker, Dr. Timothy Mercer from the Australian Institute of Bioengineering and Nanotechnology, and Dr. Pekka Ellonen, the head of the laboratory at the Institute of Molecular Medicine in Finland. Following my presentation, both Tim and Pekka will discuss their use of oligos printed with our technology. We at DNAscript are both proud and excited to announce the commercial availability of the Syntac system, the world's first DNA printer using our proprietary enzymatic DNA synthesis technology. At DNAscript, we strongly believe that synthetic DNA should be accessible by everyone. This is a critical part of accelerating innovation in life science and plays an instrumental role in powering the biorevolution. At my core, I am a bioengineer. And a critical piece in the bioengineering cycle is the ability to write, test, edit, and read genomic information as fast and as accurately as possible. We have seen significant innovation in the areas of sequencing and gene editing to enable and accelerate scientific development, but the same cannot be said for DNA writing or as well as we call it, printing DNA. The method for synthesizing DNA using phosphoramidate chemistry has seen very little change in the recent decades. And in the context of genomics, with so many recent technological advancements, decades without innovation is far too long. Our mission at DNAscript is to make DNA synthesis as easy as editing and sequencing are today. We are confident that the next industrial revolution will be driven by life science and biotechnology, and we are contributing to this revolution by leveraging nature's nanomachine enzymes. And some of them has been trained or evolved for billions of years to become highly efficient at building DNA. Like nature, our enzymatic DNA synthesis technology, or EDS, uses enzymes to synthesize nucleic acid. EDS is simpler than phosphoramidate chemistry synthesis. We basically start with a proprietary solid support and initiator DNA from which we synthesize oligo. Then synthesis occurs in a series of two-step cycles. The first step of each cycle is elongation, where our highly engineered polymerases add base with a reversible terminator group that prevents any further addition. The second step is to deprotect the base by removing the terminator group for the next cycle, leaving nucleic acid strand ready to be elongated again. And there is a video explaining that. Let me show it.
The foundation for synthesis is a resin-based solid support with initiator DNA, or iDNA, attached. iDNA is prepared with a specific cut site to facilitate oligo release after synthesis. A proprietary and highly engineered terminal deoxynucleotide transferase, or TDT, incorporates the sequence's first nucleotide to the iDNA. These unique nucleotides have a reversible terminator to ensure single base addition per cycle. An acidic reagent is then added to reverse the nucleotide terminator. After washing, the oligo is ready for the next nucleotide addition. This cycle repeats until the oligo sequence is built to the desired length. Completed oligos are enzymatically cleaved from the iDNA cut site. Then purified, quantified, and ready for use.
So EDS provides faster, simpler, and more convenient solutions for DNA synthesis. Our company's inception was in 2014, and in the 7th year since that time, we have accomplished what it took decades to achieve with chemical synthesis. Our achievements have enabled us to merge the science of electrical engineering and bioengineering to produce a fully automated solution for printing DNA right on your benchtop in your lab. In comparison to traditional synthesis, DNA synthesis on Syntax system is water-based. No specialized or highly trained staff are needed to operate the Syntax system, where every synthesis run is designed by you and initiated when you schedule it. 2021 is a pivotal year for the life science industry, and the first real change in DNA synthesis in decades is happening with the launch of our EDS technology and the Syntax system. So our Syntax platform is composed of the Syntax system, Syntax console software, and Syntax reagent and consumable kits to automate enzymatically DNA synthesis. The setup for a run takes below 15 minutes. You start by importing the sequence into the console software. Next, you load the reagent and consumables as guided by the touchscreen on the Syntax system. Then you press start and walk away. The Syntax system prints your custom oligo, then purifies, quantitates, and normalizes them for immediate use into your application. The Syntax system prints today 96 oligo in parallel at lengths of up to 60 nucleotides each. 20 oligos can be printed in approximately 6 hours, thus enabling 2 runs into a standard workday, and 60 nucleotide oligo is taking approximately 13 hours. We also offer the ability to customers our initiator DNA to incorporate a custom conserved sequence between 15 and 40 nucleotides on every oligo of the plate. The speed of EDS coupled with the Syntax system ease of use and simplicity enable you to enter a sequence and get a full plate of oligo ready to use all within the same day. We like to say sequence in, oligo out. We have designed the Syntax system to be super user-friendly, so even novice user in any lab can walk up and start a run with only extremely minimal training. And more importantly, the Syntax system allows you to control which oligo are synthesized and when. With less risk of delays, outcomes are more predictable and productivity increases significantly. With same-day synthesis, the Syntax system enables you to design, print, and test your oligo within 24 hours and even faster with shorter sequences. By accelerating this cycle, you can quickly iterate through oligo design to optimize assays faster and more efficiently. For example, in a real-world case study from one of our external evaluation partners, the Syntax system enables a design print test cycle of 9 hours. This pace enables them to optimize their assay in just 5 days. This was a significant time saving in comparison to our previous experience using oligo order from a service provider. The time saving could be even more significant with the logistical delays as seen past year due to the pandemic. Imagine how many additional projects you could complete in the time spent waiting for oligos to be delivered. When rapid results are needed, having the Syntax system right in your lab is a game changer. Before launching the Syntax system, it was important to us to have our technology and the system itself evaluated by researchers running real-world applications in the field. In total, we work with 41 evaluation partners across a diverse set of demographic and application since the start of 2020. We initially shipped a plate of oligo to test in various applications, then starting placing a beta version of our Syntax system with certain partners. In total, 9 beta versions of the Syntax system were installed, and the performance results exceed our expectations. Our goal with the evaluation program was to ensure that our Syntax system meet expectations for ease of use and performances. And more importantly, we wanted our partner to have the same confidence in the EDS-made oligo as we do. To date, over than 8,900 oligos have been sent to our partners. Here are the most recent data from our partners using our most recent kit upgrade. As you can see, almost the entire oligo, independent of their length, are above the average quality reported for chemical synthesis. And more importantly, a vast majority of those oligos are in fact above the gold standard quality of the chemical synthesis. With our EDS technology, we have achieved an average cycle yield of 99.6% of oligo designed by our partner for their own assay. This gave us great confidence to move forward with the Syntax launch. Overall, EDS oligo have been tested and validated by our partner in various genomic applications. Syntax oligos have shown substantial functional equivalency in key genetics and genomics applications, including PCR, qPCR, digital PCR, amplicon sequencing, mutagenesis, CRISPR, and variant confirmation by Sanger sequencing. And I can tell you this is just the beginning, as we are continuously making improvement to our engineered enzyme, our biochemistry reagent, and developing new methods to enable the Syntax system to print longer, oligo faster, with higher fidelity and support new application. And here is a glimpse of some of the application we are developing. With future version of our kits and technology, we will enable more application in NGS, FISH, gene expression, and gene assembly. Simply put, there is more to come for the Syntax system and DNA script. And before I close, I would like to introduce two of our evaluation partners and guest speakers who will share their experience testing oligo produced by EDS. Our first speaker is Dr. Tim Mercer from the University of Queensland in Australia. And following Tim is our second speaker, Dr. Pekka Ellonen, Head of Laboratory at the FIM Technology Center and Sequencing Unit at the University of Helsinki. And lastly, I would like to highlight that we have a poster at this conference that was prepared in collaboration with the Jackson Labs, showcasing the value of same-day oligosynthesis for COVID research in a time when synthetic oligo made by chemistry were in short supply due to high demand. The poster will be presented on Monday by Dr. Benoît Dorien, an application scientist at DNA Script. So again, the Syntax system is available today.
So thank you for your interest into enzymatic DNA synthesis and into DNA Script. If you have any question, please feel free to reach out via email. We really look forward to hear from you.
Now it is time for me to introduce Dr. Tim Mercer, who is an associated professor at the University of Queensland and a principal fellow and GL at the Australian Institute of Bioengineering and Nanotechnology. Tim received his PhD at the University of Queensland, and he is going to share with us today his talk about the synthesis of captors, library adapters that provide real-time control of nanopore sequencing. And with that, I will turn it to you, Tim.
Welcome. I'm Associate Professor Timothy Mercer from the University of Queensland. And today I want to describe to you our design and synthesis of captors, custom library adapters that provide real-time control over Oxford nanopore sequencing. Hello, everyone, and welcome. Thanks for joining me here for my talk today. Firstly, I want to also thank DNA Script for inviting me to talk today. Today, I wanted to describe some of the new things that we've been doing and building with their enzymatic DNA synthesis technology. And the project I wanted to describe was how we use the system to build custom library adapters. So we use the flexibility to build a range of different custom library adapters, or captors, as we have nicknamed them. So let me just quickly begin by starting to describe what a library adapter is before we talk about how we've customized them. So library adapters are short oligonucleotides that are ligated onto the ends of your sample DNA during library preparation. And these adapters include sequences such as primer binding sites that are essentially needed for the sequencing reaction to proceed. So these library adapters essentially are needed for you to do your sequencing. However, over the years, we've started to innovate additional functional elements that can be also added to these necessary adapters. For example, as you're probably aware, we can include unique molecular identifiers in the adapters, and this prevents or allows us to identify when reads have become duplicated. Alternatively, or additionally, we can also include sample indexes in the adapters, and this allows multiple samples or multiple libraries to be multiplexed and run on a single sequencing flow cell. Now, what we wanted to do was we wanted to develop our own custom library adapters that included a new functional element that we call the control element. So this new element, it kind of acts as an in-read sequencing control that allows us to evaluate diagnostic performance and provides qualitative and quantitative control over our sequencing. And in the case of Oxford nanopore sequencing, as I'm going to show you later, this actually allows us to actually dynamically control our library sequencing and the nanopore function. So just put a pin in that for a moment, however, because before I describe how captors work and how they achieve this, I want to just take one step back and quickly describe how nanopore sequencing works. So nanopore sequencing occurs by measuring changes to the electrical current as a DNA molecule is fed through a membrane-bound nanopore. So as the DNA passes through, it impacts the electrical current across the nanopore, and this can be detected, these changes can be detected, and the sequence can be determined in real time. Now, for the purposes of our discussion, there's really two key things about the nanopore sequencing that are important to remember. The first is that the nanopore sequencing can sequence very long reads, sometimes up to more than a megabase in length. And that means that when we designed these custom adapters, it meant we could make them quite longer. There's not really any space constraints on what we could do in terms of designing and synthesizing them. This meant that it gave us a fair bit more room to undertake new innovations within. Now, the second feature of nanopore sequencing is that the library adapter is the first part of the DNA that feeds through the nanopore. So it's the first sequence that's read. And as you'll see later, this is quite useful when it comes to real-time nanopore sequencing. So with that sort of background, let's return to our captors. So as mentioned, the captors are essentially a custom library adapter, but they incorporate this new, what we call a control element within it, shown there in red. Now, the control element is actually not that complex. It essentially comprises two sort of flanking constant sequences, and in the middle, there's what we call a variable sequence. So the variable sequence is just a sequence that is different between different types of captors. Now, what that variable sequence is isn't too important, but what we wanted to do is we wanted to represent the widest range of different sequences within that variable region. So what we did is we essentially encompassed all the different sigmas within those different variable sequences. So using the DNA syntax, we essentially synthesized a pool of 72 captors that each contain a different variable sequence. But when you look at them collectively, those variable sequences across all the different 72 captors actually encompass all the known sigmas that are available. And the reason for this is because it essentially provides us with a wide range of nucleotide contexts by which to evaluate the base calling of Oxford nanopore sequencing. And this will become, I guess, a little bit more sort of apparent in some of the later slides. So what we did is, like I said, we synthesized our pool and we combined them together into a master mix. And we used that master mix to prepare libraries from DNA samples that we'd collected from the Sun Lake, which is a lake in the crater of the Nevada Toluca volcano. It's about a nice day's hike outside Mexico City, but don't worry, it's not too active. And we prepared these DNA samples using our captors and then sequenced them on the MinION instrument. Now, the first thing we wanted to do is we wanted to take advantage of those sigmas to evaluate what our sequencing accuracy is. So here you can see there's our sort of control element sequence. There's the variable sequence in the middle, and you can see it's got all those different sort of sigmas there. And what we can do is we can measure the performance or the sequencing accuracy of each of those different sigmas. And what that means is it provides us with a very highly detailed, but also a comprehensive profile of the sequencing accuracy within our library. But also it provides us a profile of the sequencing accuracy within each individual read. So because each individual read has a captor associated with the different ends, it allows us to measure what essentially the sequencing accuracy is for that particular read. Now, we do see a fairly wide variation in the accuracy across the different sigmas, about a 33-fold. And we can essentially use the captors. We actually see a fair bit of variation between different libraries, between different library protocols, between different samples. And this really shows the sort of inherent variation in the base calling performance between the variation between different flow cells and different experiments. Now, in addition to measuring the performance of each individual read, we can actually also start to use the captors to measure the performance of individual nanopores across the duration of the experiment. So here what we've done is we've used the captors to measure the accuracy for different reads that have been sequenced by this particular nanopore, pore number N32, across the duration of the sequencing run, which is about 24 hours. So each of those dots represents a measure of sequencing accuracy by the captors. Now we can then take a step back and we can use the captors to compare performance between all the different nanopores that are on the flow cell, or more generally, the flow cell itself. So here, there's a schematic diagram showing what the flow cell looks like. Each of those circles represents a nanopore well, and the color represents the mean performance of that well across time. Now, each of those side inserts, again, shows the performance of individual pores, with each dot representing an individual read. So what this shows really is the different levels, the different hierarchy of information, that really granular resolution of data that the captors provide us in terms of measuring sequencing performance. So we can measure performance of an individual read, an individual pore, and then the flow cell, and then the flow cell at different stages within the sequencing experiment. Now, why would we want such a granular and unprecedented amount of data on our performance? Well, one of the additional features of nanopore sequencing is called adaptive sequencing, or read-until sequencing. And in adaptive sequencing, the nanopore essentially reads the initial part of the DNA sequence, the DNA molecule, and decides if it wants to, if it's interesting enough, it can decide to essentially proceed with the sequencing, and that's shown in the top blue panel there. However, if the pore decides that the sequence is not interesting, what it can actually do is it can reverse its polarity and eject the DNA sequence back from whence it came. And what this allows it to do is it selectively targets sequences of interest whilst omitting sequence or regions of non-interest from the sequencing reaction. Now, this is great, right? Because our captors are the first sequences to pass through the nanopore. So they provide an initial indication to the pore whether the read is good quality and whether the nanopore should sequence it or not. If the capture indicates poor quality, or it indicates the wrong sample, so the wrong sample index, or it has a UMI that's been already read, so it's a duplicated read, the nanopore can decide to eject it from the pore and prevent it from being sequenced. And in this way, what we can do is we can start to integrate that really high-level granular data that's being provided by the captor on each read and each pore. We can start to integrate that information with adaptive sequencing to provide dynamic control and performance over the flow cell. This allows us to balance different sample depths, to eject reads that don't meet minimal standards, and ensure that diagnostic requirements or standards are met. Now, for the last few slides, I want to describe one additional dimension to capture functionality. So in the previous experiment, what we did is we combined all the different captors into a single pool. So we synthesized them and we sort of flexibly designed the different variable sequences. But then what we did is we essentially combined them into a pool. And you can see how they provided us with qualitative information about our samples. So sequencing accuracy, these sorts of things. In the next example, I want to show you how you can also use captors to make quantitative inferences about your sample. So how we can use the captors to provide a quantitative understanding of our library. It adds an additional quantitative dimension to our understanding of the sequencing library and how it proceeds. And to do this is actually quite simple. What we need to do is we combine the captors at different concentrations into that pool. So they form a staggered concentration within the master mix. So it means that some captors will be rare, while other captors will be abundant. And then this staggered master mix is used to prepare libraries and sequence them on the MinION. And as you sequence the captors, what you're doing, as you sequence your reads as they come through your nanopores, you can actually count the captor and compare it to the expected concentration in the master mix. And what that'll start to do is it'll start to assemble a quantitative reference ladder, seen there on the right-hand side, that provides a measure of the quantitative performance of the library. So here's some examples which show us some real data. This shows us, with that particular metagenome experiment from the Sun Lake, it shows us our sequencing at 6 hours, and then the same flow cell at 24 hours. And what you can see is that by counting the captors as they come through the pore and comparing them to the expected concentration, you can see it forms this nice staggered quantitative reference ladder. Now, at 6 hours, you can see that our sensitivity, what it does is that indicates what our sensitivity and our limit of detection is there. So you can see at 6 hours, our limit of detection gets down to the sort of the blue captors. However, as sequencing proceeds and as the library gets deeper, you can see that our sequencing increases, our quantitative accuracy increases, and our limit of detection extends right down to even the rarest captors. Now, as the sequencing is happening in real time, it's quite useful because what it means is we can use the captors to assume that we want to sequence to a predetermined level of sensitivity, and then the sample can be stopped when we reach that limit of detection. The other advantage of this ladder is that we can use the linear regression calculated from this ladder. So that linear regression shown there as the dashed grey line, we can use that linear regression to convert between read counts, captor read counts, and the known molar concentration of the captors. So what it's doing is by using that linear regression, we can convert between the subjective read counts that are specific to only this library, and we can convert them to an objective unit of measurement, molar concentration, that extends beyond this single library. And what that means is it allows us to compare between libraries. So what we can do is we can use captors. This is quite important because what we can do is we can use captors and the empirical sort of standardization and universal standardization they provide to normalize between multiple libraries. What they do is, because they provide a universal quantitative reference, we can use them to normalize away batch effects, technical differences between libraries, whilst retaining bona fide biological fault differences. And this is no small improvement as well. As you can see there on the left-hand side in blue, that shows us our samples taken from the Sun Lake. This is what they look like. This is what their profile looks like if they haven't been normalized. In the middle in green shows the current standard best practice for normalization. So you can see there's an improvement, but you can see there in yellow on the right-hand side, when we use the captors, how much more accurate and improved our normalization is. And what this means is we can much more accurately and confidently identify fold differences between samples. So we can identify smaller fold differences between samples, and we can identify them more confidently. Now, beyond this particular metagenome experiment, this same concept actually extends out to any library that's been prepared using these captors. So any library that's been compared using the same pool of captors can be compared to any other library that's been prepared using those captors as well. So what this means is this is actually quite useful because it means we can start to, if we use the captors across large numbers of samples, we can essentially start to compare those libraries regardless of when and how and where they were sequenced. So it allows us to actually start transcending, if you change the reagents, if it's sequenced at different times by different people in different places, we can essentially start to use the captors to normalize those differences. And fortunately, because we estimate that even this single pool of captors that was synthesized using DNA script, even this single pool is sufficient for millions of libraries. So in this way, we can use this pool to essentially build a network of samples that can be accurately compared to each other, regardless of when, how, or where they were sequenced. And this sort of interoperability, this sort of ability to compare between an expanding network of samples will become increasingly important as we try and sort of integrate a much larger number of samples, patient DNA samples that are going to be sequenced over coming decades, and we need to incorporate them or integrate them into an expanding sort of and evolving clinical environment. So on that sort of forward-looking note, I would like to end. But before I do, I just want to thank the hardworking scientists that have performed and supported this work. Within the lab, Helen and Scott, who have done the analysis, as well as Bindu and Igor, who have prepared the captures, prepared the libraries. I also want to thank Jeff and Marike for supporting us, and DNA Script more broadly for supporting us with the synthesis of the captures and these experiments. And I also want to thank you, the reader or the listener, for joining me here today for this talk, and the ESHG Conference organisers who have provided this, this fantastic conference. So thank you very much.
Thank you very much, Tim. Let me now introduce our next speaker, who is Dr. Pekka Ellonen, the head of the laboratory at the Institute of Molecular Medicine Finland Technology Center and Sequencing Unit at the University of Helsinki. He's going to share with us today a talk about SARS-CoV-2 variant surveillance in Finland. Turn it over to you, Pekka.
Hello, everybody. My name is Pekka Ellonen. I'm the head of sequencing laboratory at FIM Technology Center in the University of Helsinki, Finland. Today, I will talk about how authorities, researchers, and co-facilities joined forces for the SARS-CoV-2 surveillance in Finland when the spread of the UK variant required action from the authorities in early 2021.
So my topic is SARS-CoV-2 variant surveillance in Finland. And here's the requested commercial disclosure information. In the late 2020, the spread of UK variant took many authorities by surprise. There were no processes in place for real-time sequence-level surveillance of novel variants. In Finland, the sequencing workflow was established over organizational boundaries. Constant development of NGS methods is needed and is greatly facilitated by rapid oligonucleotide synthesis, enabling feasible pilots. To introduce the organizations involved, I show this slide. So, Finnish Institute for Health and Welfare, THL, is the primary authority for COVID-19 surveillance in Finland, and it's also the national sample hub. Helsinki and Uusimaa Hospital District, HUS, is the largest hospital, and it serves as a sample hub for southern Finland and metropolitan Helsinki area. Department of Virology in University of Helsinki has the academic expertise and research facilities, and they are the first COVID-19 sequencing laboratory in Finland. They also have the expert NGS data analysis team for COVID-19. Institute for Molecular Medicine Finland, FIM, is a translational research institute within University of Helsinki, also headquarters for the COVID-19 host initiative, led by our director Mark Daly and Andrea Ganna. It has technology center hosting multiple core facilities such as our NGS core that provides NGS expertise, automation, method development, routines, and bioinformatics. COVID sequencing in Finland started during 2020 when the Department of Virology established some homebrew methods. The protocols were not optimal, there were some reproducibility issues, and there were limited resources to develop the method because of the excessive workload. FIM started to provide additional capacity for Department of Virology. We produced our first sequences in April 2020. There were still, for research purposes, no need for surveillance at that time. While the pandemic endured, we were constantly looking for more powerful SARS-CoV-2 sequencing methods, and we identified the Arctic protocol to be a very good match for our needs. And in collaboration with the Department of Virology, the Arctic V3 protocol was tested and validated. It's a well-documented protocol providing 100% coverage of the virus. It involves vendor-agnostic consumables. There are commercial primer sets readily available, and it supports multiple platforms such as Illumina and Oxford Nanopore. In early 2020, the sequencing efforts were quite small and scattered. So the Department of Virology had library preps in place. They had a MySeq and they had data analysis. FIM had also some limited resources allocated to this effort, and the THL was just planning to set up sequencing, but they did not have dedicated personnel to do that. So the capacity was approximately maximum of 300 samples per week. No or little coordination between units and a variety of tools involved. But since the introduction of the UK variant, DHL formally requested sequencing support from FIM, and we established this workflow where we coordinated the Arctic V3 protocol over 3 different laboratories. We coordinated barcode sets so that each laboratory had unique sets of barcodes that can be readily combined into single sequencing run. We planned fixed delivery dates for the samples. We had frequent meetings to coordinate the weekly efforts. We had weekly Novasec runs to flush out all ready-made libraries, and we had centralized data processing by the Department of Virology. So as example numbers, I showed February. So by then we were already able to process thousands of samples per month by this coordinated effort. And if rapid response sequencing was needed, we could do that using Sanger sequencing, where we have an assay for targeted S gene sequencing only, and with a turnaround time of 1 or 2 days. And all this data is returned back to DHL and HUS, and they further distribute the data to the ECDC databases and GISAID, NextStrain, etc. And this was actually quite easily implemented because we all are located pretty much on the same campus in Meilahti in Helsinki, which is the medical campus region. And DHL here, 1 kilometer away, was the sample hub for national side, and then HUS is the largest hospital serving for as a sample hub for the southern part of Finland. So we all are walking distance away from each other. Our weekly throughput was rapidly increased from 0 to over 1,000 samples per week. During week 8, you can see this all orange bar. This was the national skiing holiday in Finland, and unfortunately the team in the Department of Virology was hit by a COVID case, and they were 2 weeks pretty much everybody in quarantine. But the robustness of our workflow design was still capable of providing the services. And after the peak, we have returned to somewhat fixed amount of around 800-900 samples per week that we process through this workflow. Let's have a look in the methods. So Arctic Network is a project for addressing virus outbreaks, and they publish protocols in the protocols.io website. And this is a beautiful media for communicating this type of information. So there are multiple versions available. There are detailed SOPs and protocols. There are information about consumables and ordering information. There is interactive comments and questions sections. And we started using protocol version 4 at the time. A brief overview of the protocol is such that, of course, there's sampling, there's RNA extraction. After the RNA, there's a cDNA synthesis to provide double-stranded DNA, from which we selectively amplify the SARS-CoV-2 genome using the Arctic primers. The PCR products are then converted into NGS libraries and sequenced by Illumina in our case, and then followed by sequence analysis and downstream bioinformatics for phylogeny and transmissions. The SARS-CoV-2 amplification takes place in two different multiplex PCR reactions, each amplicon pool providing every second amplicon representing the virus genome. After PCR amplification, the product two reactions are pooled. They are entry-paired, A-tailed. The adapters are ligated and further amplified utilizing the P5 and P7 primer sites at the ends, producing a sequencing-ready library. After sequencing, we take the reads and map them back to the SARS-CoV-2 genome, and we get these nice stacks of reads representing the amplicons and resulting in ungapped coverage of the genome. In our hands, the Arctic V3 protocol performs very nicely, so we get full coverage, high reproducibility. We have streamlined the purification, so we moved out from spray bead purification for individual libraries, and we introduced filter plates that we adapted from our Sanger sequencing workflows. This is involving much less hands-on time. So our current protocol on average is plotted here with the constant orange line. So we get quite nice uneven coverage profile. But we still want it to be faster, so this is spanning at least 2 days from cDNA to sequencer, and we want to make this a true one-day approach. So we were starting to plan two-step PCR approach, where first PCR would amplify the virus genome, and second PCR is used to add the barcodes and Illumina tails. Custom oligos is needed for such a pilot, and that's when we started collaborating with the DNAscript. So the difference between these approaches is that in two-step PCR we have the locus-specific primers with overhangs that enable us to then introduce the indices and P7, P5 tails to the library in a second PCR reaction. We designed these oligos together with DNAscript, and they were delivered to us on March 8, 2021. Luckily, this protocol was already public by Arctic Network, called tailed method. We tried that, but in our hands that performed very suboptimally. So the dotted black line here shows the coverage of each amplicon. So we can see it's fluctuating quite a bit, and we also introduced some gaps with amplicon 65 to 75 performing quite poorly, likely because there's a long AT-rich region there. To tackle that, luckily, I have a collaborator, Dr. Arto Orpana from HUS, who has devised and patented a technology called heat pulse extension PCR. It's a thermocycling solution involving regular denaturation, normal annealing, but during the extension stage of the thermocycling, we go through multiple different temperatures or heat pulses. And these heat pulses help DNA polymerase to solve complex secondary structures in the template DNA. And this has a huge effect on the amplification efficacy. When we combine this with the touchdown PCR approach, we have a very powerful tool to optimize complex multiplex PCRs such as the Arctic version 3 primer set. And indeed, we tested 5 different heat pulse extension thermocycling profiles, and I show the data for the best here in blue. So the amplicons 65-75 were recovered by HPE1. We have now gapless data for all tested samples. It's still a bit low on the coverage, and we are having more experiments ongoing. But now we have a true one-day protocol. It's 2-step PCR, no purification between the PCR amplifications. We perform no normalization because we get excess deep sequencing data from the Novasec 6000 runs, even though we put up to 1500 samples in one run. The pooled library is purified only once before sequencing. So now we have a fast, high-throughput method for SARS-CoV library prep in our hands. The beauty of this is that most labs have the skills and equipment because it's regular PCR involving generic consumables. And as wrapping up, I show you a snapshot from the National Infectious Diseases Register from DHL, showing up how our sequencing workflow has enabled the monitoring of the variants, showing the introduction of UK variant into the population, the rise of the South African variant, and very recently also the introduction of Indian variant. And at the end, I would like to acknowledge all the people involved in this work. This is not my work. I'd like to thank Tiina Hannunen and Arto Orpanen for the wet lab experiments, Team THL, Team Virology, and FimNGS team for the beautiful collaboration over the workflow since the beginning of this year. And I would like to thank Team DNA Script for support for the oligonucleotide synthesis and design. Thanks for having me, and happy to take questions if time allows.
Hello, everyone. Thank you for, first I want to thank everyone for joining the session. We'll begin our Q&A portion now. We have received several questions. I'll start asking them to each of our speakers. Our first question is for Ybert of DNA Script. The question is, you say that the syntax is available now. Can you explain how much it costs?
Sure. So effectively, the syntax system is available now for selling to your lab, your group, whoever is asking the question. So the pricing can vary depending on your requirement. And because we are in a targeted launch mode, I recommend that you contact us directly through our website or through our email, contact@dnscript.com. And we will put you in touch with one of our sales representatives and get the best pricing for you.
Thank you. Our next question is for Tim Mercer. In your presentation, you used DNA script synthesis to build new library adapters for Oxford Nanopore sequencing. What other applications of DNA synthesis could you envisage for real-time ONT sequencing?
Yeah, thank you very much, and thanks for the question. I mean, I really like the idea, I guess, of combining the sort of the enzymatic DNA synthesis with the almost enzymatic nanopore sequencing synthesis. We find we do a lot of synthetic biology within our laboratory. So there's a lot of design, sequence design, designing new genes, new proteins, and so forth. And obviously combining it with the nanopore, you get this really fast sort of a really rapid design sort of iteration phase. So that sort of design and testing. So, you know, between the ability to synthesize in 1 or 2 days and then the ability to sequence within 1 or 2 days, potentially within a week, you can go from synthesis to sequence within 1 week. And that's that sort of really fast design iteration phase. And so we're certainly quite keen to put it in that sort of direction in terms of really, I guess, accelerating some of the work we've been doing in the synthetic biology field. So we do quite a lot of sort of, I guess, industrial synthetic biology. Me personally, I come from a bit more of a bioinformatic background. And so I do a lot of work with, I guess, almost like synthetic DNA controls. So these are sort of spike-in DNA controls that you spike into your sample, and they can provide internal reference controls and so forth. But one of the things I really like about the, I guess, the ability to sort of do this enzymatic sort of oligosynthesis is what you can do is you can almost design information that you want to sort of encode within your patient library. So to give you an example, if you want to make unique identifiers for a particular patient or a particular project, what you can do is you can synthesize them. You can actually record that information in the DNA using, as you probably appreciate, there's a lot of tools out there for encoding sort of ASCII information in nucleotides. So you can kind of imprint it almost internally within the library. And what that means is when that library then gets sequenced, it means that that FASTQ library, that library data set will forever have that DNA information imprinted it. And I kind of gave the sort of the simple example in terms of like sort of patient identity, because that's quite useful for things like, patient custody, storage, these sorts of things. But within the lab, we're kind of, I guess, pushing it a bit more sort of creatively, whereby those internal synthetic DNA oligonucleotides, they almost act as sort of an internal software. And what it means is the external bioinformatics software can go in there, it can almost read the information from these synthetic spiking controls. And so it can know, all right, this is a patient sample that's done cancer sequencing, and we can see that the depth has gone to this particular depth, and this is the sequencing accuracy, this is the quantitative performance. And essentially what it means is the bioinformatic tools can almost respond in their analysis to the actual information that's encoded within those libraries. And so for me, I think it's really sort of fascinating that ability, that sort of flexibility and that ability to really rapidly make information and put it into a sample, for me is really exciting, because it's almost combining, your DNA with your external bioinformatics, and it's almost making a sort of a seamless integration between the internal information encoded within the sample and the external information that's analyzed by the bioinformatics. So for me, that's where we're sort of pushing these applications over the next, at least in the short-term future. So thanks.
Thanks, Tim. The next question I have is for Dr. Pekka Ellonen at FIM. Have you adapted the new Arctic V4 primers into your protocol?
Thank you. Yes, indeed. We are in the process of optimizing the protocol for the novel V4 set of primers. And of course, there were quite many primers that changed from V3 to V4, so that takes us a few steps back, but we are working on the heat pulse optimization to get the coverage as good as it was for V3 and hopefully even better.
Thank you. We have another question for Dr. Mercer. What would you do with the benchtop ability to synthesize DNA in hours? Which research, clinical, or industrial applications do you think this will transform?
Hello. So yeah, I mean, look, it's a pretty interesting question. I mean, I guess previously I sort of described, particularly in terms of using the sort of the synthesis, the DNA synthesis in combination with the nanopore. I think if I was sort of to take a bit of a step back and really talk about like where I think the long-term potential and the really sort of interesting questions are, for me, it's certainly in this ability now, we're starting to get to this ability to actually start designing de novo genes and de novo proteins. So this ability to essentially start, if you think of the sort of the natural history of life and evolution, all of our proteins, all of our compounds have been based on sort of previous genes that have been slightly changed and slightly modified. Whereas now, with this ability to essentially do DNA synthesis and generate our own genes, it means we can explore a much wider, diverse landscape, a much wider sort of informational space. And for me, this idea about basically being able to design de novo new genes, new proteins, even new genomes, and synthesize them and test them and tailor them for specific requirements, I find that sort of concept, that idea really, really exciting. If you think 4.5 billion years of evolution has got us here, and there's a huge amount of like the compounds and enzymes and the capabilities are massive. But now we're actually kind of moving into this new phase where we're actually no longer sort of, I guess, encumbered by what's been previously evolved, but we can start to design it ourselves. And I think, you know, we've still got, this is if the last term was like sort of short-term applications, this is more like a long-term view. I think we've got a long way to go in terms of things like the computational side of things, the design, the optimization, building these sort of screening libraries. But I do think, you know, we've seen even recently the things like with the AlphaFold in terms of being able to predict structures, the ability to start predicting structures, design them, designing backbones. I think it's a really exciting time in terms of being able to build entirely new biological compounds. So for me, I think the long term, and that's obviously going to have big implications, both research, but obviously clinical in terms of the design of new drugs and so forth. So for me, I think that's really one of the other exciting applications. There is another one, and maybe if we get time, we can go to the other one. I also do a lot of work, I mean, as many people probably appreciate, with things like mRNA vaccines and RNA therapies, which have really sort of come to the fore in the last year. And so we're also, in terms of sort of optimization and the rational design of these, this sort of like novel sort of RNA therapy class, we also see a huge amount of potential there in terms of teeing up our sort of DNA synthesis capabilities with those capabilities to make large sort of RNA compound libraries and screen them and understand, you know, what can have an impact within the cell and what can be sort of used, rationally designed to form these this sort of novel class of drugs. So thank you.
Thank you, Tim. Another question for Dr. Ybert. This is regarding the syntax system. How flexible is the system? Can you do partial plate synthesis? Can you print many different sequences in a single run? Can you print the same sequence many times in a run? Tomas, you're muted.
Yeah, thank you for the question. I think it's really interesting to learn about the flexibility of the syntax system. So basically, our kits provide enough consumable and reagent to perform 6,000 cycle kits. So that means that 6,000 nucleotides to synthesize. And those kits could be used to print oligo in a 96-well plate in 1 or 2 separate runs today. So with that means that you can easily print a full 96-well plate of 60 nucleotide oligo, or for example, 2 96 full-way plates of 30 nucleotide oligo. And with our console software, you can design run where all the oligo have the different sequence or the same sequence. So pretty much you have all the flexibility you want. And if you, for your particular application or your particular workflow, you think that you need more flexibility, you can always talk to us, talk to our team, and the system is really flexible. So we could accommodate for additional flexibility if you want.
Thank you, Tomas. And another question for you. Can you use the syntax system to build genes?
Yeah, so that's a very good question. So we are not currently marketing the syntax system for gene synthesis. The oligo produced can certainly be assembled into genes, fragment, or for gene synthesis workflow, and we have an ongoing project to improve our synthesis performances and our product to be better at meeting the need for synthetic biology market. But depending on your current workflow and requirement, the synthesis system available today may be the solution. So I can encourage you to reach out to us through our website to learn more. While the oligo products can certainly be assembled into gene fragment for gene synthesis workflow, we have again an ongoing project to make that. So the final quality of the gene assembly may differ from what you can get from the current supplier. So that's why we are continuously improving our biochemistry, and that's why it may or may not fulfill your requirement today, but we're making progress, and our goal is to be able to do genes with the syntax. So again, for current status of the performances, I encourage you to reach out to us.
Thank you. And I just want to say that we're wrapping up the Q&A session. I want to thank our speakers and thank all of you who submitted questions.